1.
This is TWIS, This Week in Science, episode number 725, recorded on Wednesday, June 12th, 2019. Kidding about science. Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with worms, elephants, and the moon. But first...
Disclaimer, disclaimer, disclaimer. You know, they say that the children are our future. But in order for that to be true, there has to be a future to begin with. I'm not surprised when children get upset hearing about the world that they will be left. So in order for us to nurture the curiosity and promise of what could be the greatest generation yet, we have to do our best to right wrongs, set the stage, and leave nature, beauty, and creativity for those generations to come. The first step preserving this future is to foster that same curiosity by encouraging questions, celebrating individuality, and setting the stage for wholesome education that is accessible to everyone. Let's start all of that by exploring this week's discoveries on This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Kai.
And a good science to you too, Blair. Welcome, everyone, to another episode of This Week in Science. We are back again, and we are Justin-less. Well, actually, we have a miniaturized, not Justin.
Yeah.
With us tonight is my my son, Kai. Say hello.
Hi.
Hey, awesome. He wanted to join us tonight, and I was like, this sounds like fun. So we hope that we will all have a really good time tonight. Justin is on his way to Albuquerque, Santa Fe. He's laying the groundwork, getting there before us. We will be seeing him this weekend at the show at the Interplanetary Festival on June 15th, Saturday. We are very excited and hope that we'll be able to bring it to you through live streaming. You excited, Blair?
I am so excited. I'm definitely feeling a little bit out of my element. I started preparing and I was like, none of these animals have anything to do with space. But I'm very excited to learn a lot of new things, which is what this show is all about.
Exactly. It's all about learning. Kai, are you excited about anything for Santa Fe?
Well, I'm really excited to go to Meow Wolf.
Oh, Meow Wolf, yes. It doesn't have anything to do with space, but it's going to be cool art.
I'm going to have tons of burritos, and quesadillas, and tacos.
That's great.
Sounds great.
Excellent.
Gotta have some good goals there. All right, everyone, we are ready to jump into the show. I brought stories about worms, our ancient ancestors, and blood, because I do love the blood. Kai, what did you bring? What story are you going to talk about?
I'm going to talk about the mass anomaly on the moon's polar south pole.
Okay, we're going to talk about that in a little bit. And Blair, what is in the animal corner?
Oh my goodness. I brought mathematic bees. I brought elephants. I brought twin guts. And I brought feather lice.
Feather lice?
Feather lice?
Ugh. It just makes me, all those feathered accessories, it makes me a little worried.
Okay.
Oh my goodness, you just wait.
Lice rule the world.
Don't share feathers with friends. Okay, and as we jump into the show, I want to remind everyone that you can find out information about the show at TWIS.org. But if you're into this podcast thing, you can find us and subscribe to us all places that podcasts are found. Stitcher, Spreaker, Spotify, Pandora, radio.com, TuneIn, iTunes, Google, all the places. And now let's jump into some science.
Ooh, let's do it.
Yeah, we love the science. So to start, because my offspring is here this evening, I thought I would talk about offspring. Well, how we as parents pass things on to offspring. I mean, the study wasn't done in people, so it's not exactly applicable, but worms, we use them as a model species for all sorts of genetic things. And so researchers have done it again, looking at Caenorhabditis elegans. C. elegans, if you're going to. Yeah, say it a little bit more shortly. Don't fall out of the chair with so much excitement, Kai.
You're just so excited for the worms.
I know. So there were a couple of papers published this last week on the passing, not just of genetic information, but of behaviors from one generation of worms to the next. And the thing that's really cool about these two studies is that they are really starting to get at a mechanism of how behaviors learned during an organism's lifetime, like to avoid a predator or a fear response to a particular stimulus, how that can potentially be passed on to offspring and last over multiple generations. And so one paper published in Cell earlier this week was out of Tel Aviv University. They used C. elegans, and they demonstrated that small RNA molecules, these little RNA, these small interfering RNA molecules, get transferred from one generation, from the adults to the offspring, that allows a changing of how genes get transcribed, and that changes the way that behaviors are expressed in the offspring, and it can last for multiple generations. Another research lab out of Princeton University observed that bacterial species can be avoided by these worms. So C. elegans, before they die from a pathogenic bacteria, they learn avoidance of them. Some kind of stimulus from these bacteria leads them to want to avoid them, and then they're able to, again, use these small RNAs to pass on that ability from one generation to the next. The Princeton group, they were really interested in how these worms exposed to a bacteria had a higher survival rate compared to young that had been born from unexposed parents. And they discovered that there was a gene called DAF7 that is actually linked to pathogen avoidance behavior in C. elegans. After the worms were exposed, DAF7 got upregulated. The expression of this gene was increased in a subgroup of neurons, and it stayed elevated 4 generations. So it was learned, one exposure to the bacteria occurred, and then this behavior persisted for 4 generations. So one of the big questions coming out of this is, why would a behavior like this avoidance behavior, last for so many generations? And one of the ideas that they've come up with is that these worms don't live very long in the scheme of things. They're not long-lived organisms like we are. And so the bacteria, the pathogens, are probably around for the similar amount of time. And some of the bacteria may actually be beneficial. So it could have been just kind of a chance meeting, or maybe certain bacteria became pathogenic in a particular situation, but then the pathogens died back and the beneficial bacteria take over. And so after several short generations down the line, you don't necessarily want it to persist indefinitely, but you do want it to be a persistent avoidance to help with that ability to survive. And then the other study that came out of the Tel Aviv University, looking at these small interfering RNAs, these sequences that can silence genes, they bind to messenger RNA transcripts, and by binding to the messenger RNA, which then goes to transcribe genes, what it does is it silences the messenger so that the gene never ends up expressed. And these were transferred from neurons to the germline. So these were behavioral neurons in the worms. They produced a protein that's called RDE4, and it had to have these small interfering RNAs in the nervous system, and they get generated alongside the RDE4 in the neurons. They don't get generated anywhere else. The question is, how did these RNAs go from the nerves to the germ cells? But we know that they are involved.
Okay.
And so there's some really interesting questions going on here.
So I have so many questions.
I know.
Me too.
So the first thing that comes to my mind is, we've talked before about genetic basis of behavior. We've talked about epigenetics. We've talked about genome editing. We've talked about all these different things. This is a weird kind of Venn diagram of all of these different concepts.
Yeah.
And so my mind can't help but go, mostly just because I'm obsessed with corvids, but to that story about how there were people that wore a particular mask and treated crows badly. They would throw rocks like not directly at them, but near them, or do something to disturb the crows. And then generations later, they would avoid people in these masks. So the previous assumption was that there was some sort of communication happening from generation to generation. Obviously, this is a way bigger behavior thing than just being able to avoid a particular bacteria. But this is, if I'm going huge, big picture on this, if behaviors could be genetically influenced, there could be assumptions that we're making in the biological world that are wrong. If there are genetic bases to some of these behaviors.
Yeah, and then the question is, at what point does something go, like, we know there are lots of innate behaviors, things that animals just do, right? And the question is, how do they go from being learned behaviors to innate behaviors, something that's a reflex, something that just happens when a certain stimulus occurs? And so is this part of that process? At what point does it go from these interfering RNA molecules, these little tiny snippets of RNA that either affect messenger RNA or like actually bind to the genes to upregulate or downregulate their transcription? But then how do they go to actually being encoded and changing the behavior? And so that is something that eventually happens. So sometimes the behaviors, the RNA goes away, right? Behaviors go back to the wild type. Or the RNA maybe gets inserted, something happens, and the behavior changes forever. Like, so this is, to me, we're starting to get at these mechanisms like you're talking about. How does this persist over generations? What does learning really mean?
Yeah, and when we talk about animal instinct, is this stuff that is in their genetic code? That a wolf has to be raised by a mother wolf to learn how to hunt and be a wolf, but an alligator pops out of the egg, knows how to be an alligator. So is there a genetic difference there?
Right. So, and it just adds,
really
we knew epigenetics is complex, right? And how it moderates and modulates our genetic code. But now this is like this extra layer of complexity.
This is
like,
fascinating.
It's
a treasure trove. It's a treasure trove of research. Hey, researchers, get on out there.
Just get out there. Kai, do you have any thoughts about all this?
Yeah, well, mostly hers, but mostly Blair's. But one of my one thing that I found really interesting
Mhm.
was that how like it basically makes an RNA copy of it after it gets attacked, and then whenever it gets attacked again, like throughout like 4 generations or this generation if it survives
Mhm.
and lives on, it will have a defense against it.
Right. It'll avoid it. Yep. It learns it and it gets encoded through the RNA. So the RNA is changing. That expression of those little RNA molecules is changing. And what makes that persist? Yeah, good question, kiddo. Good question. Moving on from worms to sponges, because let's go even further back in our evolutionary history. Why don't we? Apparently, there is a story out of the University of Queensland this week that may rewrite evolutionary textbooks. Or
Oh, give it to me. I love it.
yeah, let's get it. Let's get in there. Look at you drinking with your little metal straw.
Oops. Fully.
How ecologically and environmentally conscious are you? Yeah.
I try.
So this study, scientists at the University of Queensland decided they wanted to go back and look at this idea of who really was the multicellular organism's ancestor. Where did our multicellularity come from? And what did you learn in school, Blair? When did we become multicellular? When did that happen? Who were the organisms?
So that was like one cell like ate another cell, and then
they formed a union, and the bigger cell provided shelter, food, while the other one uses free stuff to create more stuff that's harder and more complex to get, and made it more efficient.
Right, but the oldest, like the organism, when you take basic biology, is like the precursor, right? Not just archaebacteria, but when you're talking about multicellular life, sponges. These are the old organisms. And so researchers went, okay, at the sponges, multicellular. Yeah. Well, look at this other organism that are known as, so there are these multicellular sponges, choanocytes, but the cells within these sponge choanocytes look a lot like single-celled choanoflagellates. They're single-celled organisms that have flagellum, but they based on the way that they look, they lumped them into the same group and said these single-celled choanoflagellates must have turned into the multicellular sponges where their choanocytes are all working together. But they took a look at that assumption, at this idea that we've always just taken as single-celled to multi-celled, and this is how it worked. And they compared the transcriptomes of these sponge choanocytes and the choanoflagellate single-celled organisms. And what they discovered is that they're not the same. They're not as closely, they have different transcriptomes. The sponge has all sorts of instructions in its genome, the transcriptome, the things that get transcribed, those proteins that make it what it is. And the idea is that they're looking at it, and the researchers say they're looking for signatures, these cell types that they're like, if they evolved along a lineage, then their signatures should be similar in the transcriptome. But when they looked, they were not. The researchers say these biologists for decades believed the existing theory was a no-brainer. Sponge choanocytes look like single-celled choanoflagellates, but their transcriptome signatures simply don't match, meaning that these aren't the core building blocks of animal life that we thought they were. And so by looking at the transcriptome, they say they're taking a core theory of evolutionary biology and turning it on its head. I mean, at least a page or two of evolutionary textbooks will have to be rewritten.
It's like page 1 and 2.
Yeah, and so the interesting thing is that the signature for multicellularity really is one of differentiation. And so what they hypothesize now is that instead of these choanoflagellates, is that there was a sponge precursor that had more stem cell-like characteristics, where it was a cell, instead of being differentiated and saying, I'm a choanoflagellate cell, it was a cell type that was able to switch and change and turn into supporting cells that would allow multicellularity, where cells take on different roles, as opposed to just being a colony.
That makes sense to me. I mean, overall, the sponges, when I brought my sponge story, I think it was last week, the very first thing I said was, a lot of people don't realize sponges are animals. But I think that, you look at a sponge and you think it's pretty simple, but when you look at a diagram, yeah, it's pretty simple. But they have different parts of their body. They have this differentiation that you're talking about. It makes perfect sense to me. In some ways, I would expect that other multicellular organisms like plants, those seem more like something that's just a multiplication of a similar cell, because, you look at those under a microscope and they look kind of the same. They're like they're little bricks.
Yeah.
But I mean, it means there's a lot of unanswered questions out there for sure.
I know. I mean, and this is like such an assumption that this forms this basis of how we have thought evolution took place. And all of a sudden you look at it a different way, through a different lens, and realize, oh, we characterized things wrong. Kai, you had a comment.
Yeah, it's actually something that's actually kind of like one of those, like, that take different roles. The Portuguese man of war.
Oh yeah.
There's different cells put together into different clusters combined, making different like animals combining together, creating like stingers, which have jobs, and then things that eat the food after the stingers kill them. And then there's one that keeps it buoyant, that just looks like a gyoza. It looks like gyoza.
Looks like gyoza.
Yeah, actually it looks like purple gyoza.
Don't eat it though.
Don't eat the pot sticker of the sea.
Yeah.
No.
Bad idea. Do not do it. Do not do it.
Yeah. So I mean, to Kai's point, that's a perfect kind of next step, which cnidarians are after the sponges. So you have this kind of specialization in an amorphous blob of a sponge, and then you have the specialization of cells within a progressive organism, which is totally weird.
Yeah.
It's pretty neat.
Yeah, it's a big question. How did this happen? Anyway, this paper from the University of Queensland, primary author Bernard Degnan, published in Nature, Pluripotency and the Origin of Animal Multicellularity. It looks like it might be a very interesting read for those of you who are so inclined.
It's a little bit of light reading about multicellularity.
Yeah, a little bit of light reading about evolution and ideas being turned on their head. All right, Kai, you were going to tell us a story.
Yep.
Oh, you can leave that right there. Oh, nope. Okay, just put it down. What are you doing? Oh,
It makes it I can barely see it. I couldn't see anything.
Oh, he needs to see his
I couldn't
notes. That's
see
very
anything.
important.
Yes, otherwise I couldn't read it.
I need a screwdriver now. What is going on? Okay.
Uh-oh.
Microphone.
You're not looking.
Yeah. Oh, we've got microphone issues. I always tell Kai not to touch the microphone.
I got an
This
idea.
is why.
I'll just take off the top microphone bit.
Nope. No, you won't.
And I can just talk in that.
No, you won't. I don't know.
I think we got it. Bear with us, some technical difficulties.
Okay, Kai, can you read from there? Okay. Scoot forward. Put your head in here. Here we go. You want to sit on my lap?
A moment. There.
Oh, look at you there. Okay, we can't see your face, but that's okay.
So, wait.
Okay, what were you going to tell us about what happened in the moon?
Mass anomaly that of this like mass bunch of mass found on the South Pole crater.
Right. So who found what is going on when you say mass anomaly? A bunch of mass found in a crater, the South Pole of the moon.
Well, there was this thing of there's this like iron thing that was found that was about too big for it to come up to all come from the moon, and it was like this just giant glob of iron that wasn't supposed to be there inside a crater on the South Pole, and it was about. I'm lost.
What are you trying to remember?
I'm lost.
What are you trying to remember?
Well, there's this ancient yeah, I'm kind of lost. Come back to me later. Okay. I'm having a bit of technical difficulties.
You're having technical brain difficulties?
Yeah, my brain is cut off.
Okay, so let me ask you the questions then. So, do you remember what the things were that discovered this mass anomaly?
One was Lola. I forgot the name of the other one. Grail.
Right, and what were they looking at on the moon?
This big mass anomaly of iron from an ancient meteor of a decent size that hit the moon, and they think that it was either two rocks hitting each other, and then this rock, this like space rock made of iron, hit this other one, and this one stayed there and destroyed the other one, making a bigger crater. Because there's like this really big crater, like really big one.
Right. This Aitken Basin is a very large crater, and it's a point of interest for lots of people. And if it's an asteroid, a big chunk of iron from an asteroid, what can that tell us if we go there and we learn? What can we learn about it? Do you think we can learn how craters are formed?
Yeah.
Yeah?
Probably.
And if it turns out that it's not from an asteroid, what does that tell us?
It was probably from the core of the moon.
Right. And would that maybe mean that there's some kind of something happening in the moon that we didn't understand before?
Like maybe maybe it's not cooled off and it's tectonically active or something like that.
Right. But the big question is like, why would there be this if it's just something from inside the moon, why would it have all of that mass collected in one place?
Good point.
Right.
Why would there be that much mass in one point?
Which
is why it's really confusing.
So we knew about the crater, right, before, but we didn't know about this deposit of iron?
Yes.
Okay, and so we think two things hit on the moon around the same time? Or
it could
one of
have
the
been
things was the moon and one was another thing?
Yes.
Okay, and so we have had this assumption forever that the moon is just kind of dense, solid, kind of dead, right? But it's also possible that this came from within, and therefore things are still moving in there. Which it's locked in orbit, right? So that then it might make sense that things might pool kind of unevenly because it's constantly moving.
Yeah, the interesting thing is this is on the far side of the moon on the South Pole, so we can't see it from Earth. If you were thinking of it as if it is something that is a lot of mass, means that it is an element that's collected, or a bunch of elements that are collected there that are mass heavy, which would indicate something like iron, like Kai has been saying. And so you think about iron, and then you're like, maybe the Earth's magnetic field has pulled a bunch of iron to collect in one part of the moon. But it wouldn't really make sense for it to collect on the far side. You would think that the magnetic field of the Earth or gravitational forces would pull stuff closer to us. So
what if
it is internal processes? It's just kind of weird.
What if this is a relic from when the moon broke off of our planet?
Oh, that's another idea. Interesting.
That's a good one.
What if, there was like this pool of molten iron, or the bit that broke off the planet, or the part where the meteor hit the planet and fused and broke off of our planet? What if one of these scenarios involved an uneven mixing?
Right. I mean, these are great questions.
Sounds like we might need to ask some of these questions on Saturday.
Sounds like we need to go and actually get some material from the moon.
Yeah, I have a great idea. Let's go. I heard there's an Interplanetary Festival happening this weekend. Let's go there and ask about it.
Let's do that.
Oh,
Let's find. That's right. We're going to find
No
ourselves
way.
a moon scientist,
No
a geologist. We'll find
Yeah.
somebody, a space geologist, to tell us all about it.
Yeah.
One
Yes,
moment.
yeah.
I'd also like to say that there's more than just iron and heavy metals. There's gold. There's
Mhm.
what could I also do? Copper,
Yeah, there are lots of heavy metals.
rust, maybe when combined with iron.
Mhm.
Rust. Iron rust. That's a heavyish metal.
Mm-hmm. Well, rust is an oxidized metal.
Yes.
Something oxidized, yes. All right. Thank you so much. Thank you for telling us about the mass anomaly.
You're welcome.
What does anomaly mean? Do you remember?
No.
It means that it's something strange and unexpected. An anomaly. It's not normal with your data. It's anomalous.
Not supposed to be there.
Not expected.
An
That's
ominous anomaly.
Anomalous anomaly.
Say that 10 times fast.
Yeah.
Anomalous anomaly.
You don't have to.
Oh, man.
That is a band name if I've ever heard one. I have not updated my band name list in years. I'm adding it.
Let's do it. Ominous anomaly.
You can't even say it.
I can't even say it. The band name that nobody can say.
Anomalous anomaly.
But
I can say it.
as we're moving forward, hey everyone, this is This Week in Science. Do you know what time it is?
Oh, what time is it?
I think it's time for Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Thank you. I was like, uh-oh.
My cue.
My brain broke. So you know whose brains aren't broken? Bees.
Bee brains aren't broken.
Bee brains aren't broken. Go ahead and don't say that 5 times fast. Okay. Bees, as we know, they are very good with numbers. We've talked on the show about how bees can tell relative amounts. They've even recently been tested to show potentially the quantitative value of 0. So they have a good reason to be able to do that. They also have good language abilities. We've talked a bunch about their waggle dance and how they can actually signal direction and distance of nutritious flowers to other members of their group, of their colony. And they can interpret that dance to know exactly where to fly. So it is no surprise that they were the most recent target for a study on matching symbols and numerosities. Amounts is basically, it's a sciencey word for amounts. So they were used as the latest one to test this theory. There have been other animals in the past that have shown success with the symbol versus numerometry. Chimpanzees, rhesus monkeys, pigeons, and a single African gray parrot. That's right, our buddy Alex. They have all been able to associate either Arabic numerals or English names to actual numbers. So already the idea that our understanding of math and number symbols as being uniquely human is kind of falling apart, but researchers wanted to see how far this went. So in this study, it's pretty interesting. They took honeybees and they had them match symbols. They called them, for the course of the study, signs, and number amounts called numerosities. They used a subset of symbols previously used in training pigeons, so they know that they're they can differentiate, the pigeons can differentiate enough between these different symbols. So they were well tested. They didn't want to use actual numbers, probably for a bunch of reasons, but one of them I would assume is so that they don't accidentally tip off the bees by something that they are doing. So it's a little more blinded to the researchers. So bees were trained to fly in a Y-shaped maze. Inside that maze, they would look at a stimulus. The bee would then fly into a decision chamber where it would view two options, one correct and one incorrect. So they were trained to match a sign to a numerosity, a number symbol to an amount, while a second group was trained to match the opposite, an amount to a symbol. If the bees were learning to match a sign to a numeral, to an amount, they would first see the sign and then have the option to choose shapes or amounts. If the bees were learning to match a numerosity, an amount to a sign, they would first see the number of items, like 3 squares, and then have the option to choose from 2 signs. And Kiki's showing them right now on the video stream. The examples they show is an N and an upside down T. And then the numerosity they're showing is 2 circles or 3 squares. So because they're different shapes, they have to understand the relative amount despite the different shapes. So if, for example, if a bee viewed an N, that bee would need to choose a display presenting 2 items. In this case, N is the symbol for 2. She would need to be able to do this regardless of shape, pattern, or color. If she chose correctly, she would get a sugar solution, delicious. But if she chose incorrectly, she would taste bitter, quinine, which we've talked about on the show before as being this kind of, it's just a bitter tasting thing. It's not pleasant, but it doesn't hurt them. It's almost
quinine,
like
quinine. It's the ingredient in tonic water.
That makes sense. It's also, I think, in some more natural bug repellents because bugs don't like the taste. It makes it bitter. Bitter. Yep. So neither the what do you say? Quinine. I've always read as quinine. It's
I called it quinine.
Quinine, nor the sugar. Neither of these are able to be smelled by the bee. So their only cue is the visual cue. So they definitely would not be able to cheat in any way. They were trained for 50 trials. They matched the signs to the numbers. And they had an accuracy of about 75%, which is pretty good. This is the first time symbol matching to number has been shown in an invertebrate. So all those other examples before are kind of those like quote unquote higher level organisms. They have a much more developed brain. So that we kind of wanted to see how far back this starts. After training, they were tested in several conditions with new patterns, new colors, new shapes. They continued the preference at about the same percentage, 75%. So it seemed that they really did learn these number symbols. But where this gets interesting and where I kind of call some question to the interpretation of this experiment is that when they tried to reverse their tasks, they did not do well. If a bee learned to match a sign to a numerosity, they could not match a numerosity to the sign, or vice versa. So the association between number and symbols was only learned in one direction. They do argue that in children, this directional learning outcome called operational schemas are common. And so you kind of teach a one-way learning process and then you have to reverse engineer. I think that's one of the reasons that word problems are so hard for students when they're learning math, is because you're trying to make them do things backwards, and it can be problematic for their learning kind of directionality that they're used to. So the question I have about this is, how do you really truly know that you are not just teaching basic association and you're developing something akin to a language skill, which is really what they're asking here. They're identifying math and numbers and symbol-based numbers to a language, a universal language that humans use. And so at what point are you just associating a visual to something else? And at what point are you developing a lexicon? Which I recognize might sound a little bit nitpicky, but I think that if you're trying to extrapolate this into a mathematical concept, you have to understand it a little bit better than that. So if they can say when the N and the upside down T are in the same circle, then they have to find the 5 objects that are together, right? Then you can start to understand what those symbols actually mean. So I wonder, maybe that's next, who knows? But I wonder how well this actually tells us. If they can't reverse engineer it, how well is this really proving this universal language of math is capable by bees? But they do understand 0. So I feel like also I kind of want to give the bees the benefit of the doubt here. They probably got it.
Yeah, and it is this interesting question of, what are the symbols and which are which are the symbols that represent what? And we're trying to enforce something that is a human ideation, a human concept onto an insect cognition. And so the question is, as we're doing all of these studies, okay, one works one way, one doesn't work. It doesn't work in the reverse. What does that mean? Does it mean that we tested them appropriately and their cognition can't support it? Or does it mean that our test was inappropriate?
Right. And that's always the question, right? I mean, for so long, we thought that monkeys, the kind of old world, the lesser monkeys, the more primitive monkeys, whatever you want to say, that probably none of those words are correct. We thought that they couldn't recognize themselves in a mirror. And it's because we developed a test that was not relevant. And now all of a sudden we found out they can.
Yeah. And then you put in the right test, the animals are able to do this thing that we've been looking for. And it's just a matter of asking the right question in the right way. And so, and then the question is, bees don't really count. They're not going 1, 2, 3, 4, 5 flowers. Yay.
Yeah.
That's not happening. There is some kind of numerology, obviously, because we're able to find these concepts that match 0, the 2 boxes, 3 boxes, but there are concepts that are equivalent, but that equivalence is not the same.
Right.
There's something else going on in their cognition about what they're actually comprehending.
Right. They don't need to know exactly how many flowers there are. They need to know that the nectar in this field is sweet and abundant.
Yes. It is sweet and abundant.
Yeah. What do you think, Kai? What do you think of the counting bees?
It kind of sounds like we're torturing bees. We're trapping them in mazes and making them do things, and if they get the answers wrong, they get gross stuff poured all over them.
Yeah.
It sounds like torture to bees.
Well, I feel like you could
Sorry, bee, you get the quinine.
Yeah. Most research, if you boil it down to some really harsh terms, probably doesn't sound great. Also, a lot of things that we humans do probably sound like torture if you explained it to another organism, right? I like to put on really tight clothes, go into a hot building, and run for an hour, but never get anywhere. It sounds like torture, but it's the gym. Yeah. So, it's all a matter of perspective, right?
Yeah.
Speaking of counting animals, we went from the very simple brains in the bees to the very complex of elephants. Elephants are some of the most intelligent animals, as far as we can tell, in the animal kingdom. They have very intense cultures, and they have their own types of language. They have amazing memory, as the sayings go. And so elephants, we know, are pretty smart. An international team of researchers found that Asian elephants, are smart in a whole nother way. The nose knows. They can tell which of 2 food sources has more food in it by smell alone.
Oh, wow.
Yes. This is
I'm just trying to imagine, you go into a pizzeria and you're like, this pizzeria has more pizza than the other.
Yes.
I know you have lots of tacos.
Yeah. Imagine
because I smell them.
Yeah, imagine somebody put like a bowl of spaghetti, fresh spaghetti with meatballs on it, or red sauce or cream sauce or whatever you prefer. Put a small bowl, piping hot. Put a larger bowl right next to it. A box over both of them with a slit for you to smell. Do you think you could tell which one was bigger? The answer is you can't.
Do you think you could, Kai?
You think you could? We should test it.
That makes sense.
Let's test it in Santa Fe. Let's get a small burrito and a giant burrito. You'll close your eyes. You can tell us which one you think is bigger. But in the meantime, as far as this international team of researchers are concerned, they were looking at an aspect of odor detection that, as far as we know, Kai could be the exception. As far as we know, no animal, including humans, has ever been known to discriminate between food portion size using only olfactory senses. Their research process was quite simple, but very effective. They took 2 buckets. They filled them with sunflower seeds. Sunflower seeds, I did not know this, are an elephant favorite. They locked the lids and had small holes on them. You got to lock the lids because elephants are very, very smart and very good at opening things. They put small holes in them and then allowed an elephant to sniff both of the buckets. They then loosened the lids, unlocked them, presented the buckets again, and then they saw which the elephant chose to open and dive into first. They repeated the experiment many times with different elephants, varying the amounts of sunflower seeds in the buckets. So sometimes it was a very vast difference. Sometimes it was a more subtle difference in the amount of sunflower seeds. The elephant showed a clear preference for the bucket containing more seeds, even though they never saw what was inside before they opened them. They also found that the greater the difference, the better the elephants were at picking out the best. So they could usually pick out the greater one, but it was much easier when there was more of a difference between the two. They also found, interestingly, that the males were better at determining quantities than they were the females, which makes sense because males are extremely territorial. They have male-based, harem-based structures. So they'll have one male and a bunch of females, right? So they have to be able to sniff out males from a very, very far ways away. So it would make sense, have better olfactory systems than the females. This is the first example ever documented of an animal being able to differentiate between quantities by smell alone. They also note that this may prove useful in Thailand. Now, this is where this gets really interesting. Aside from just, whoa, elephants are so cool. In Thailand, elephants are a nuisance and a pest. Yes, you heard that right. Elephants are a pest. They encroach on human-occupied areas. As you might imagine, if an elephant showed up in the middle of my commute to work, I would definitely be late, and that would be frustrating, although I'd probably be more fascinated by the fact that there was an elephant in San Francisco. Up until now.
Here in the US, the only time you run into an elephant or a giraffe in your path is at a wild animal park where you get to drive through it.
Yes, that's right. Up until now, most of the efforts to deter elephants in these cities have been based on attempting to scare them away. They'll use firecrackers or loud noises or stuff like that to kind of scare them off, shoo them off. But if they can find a way to lure elephants away from areas where there are lots of humans with bountiful food, they might be able to convince them to stay away from those busy areas. So that's definitely an interesting application that I never would have thought of, but it makes perfect sense. You can't really put a sign up that says elephants this way, but if you put a nice big pile of sunflower seeds in whiffing range, that might be enough to pull them out away from civilized space.
Huh. Yeah. Look at all the sunflower seeds I have. Psych.
No psych. It would actually be there. But if you think about it, sunflower seeds are much, much less expensive than firecrackers and much less expensive than disrupting entire city traffic flow.
That's true.
Yeah.
It's true. Maybe we can use this. But I mean, I think, yes, it's interesting that we can potentially take this new knowledge and put it to use. But the idea that elephants ecologically would be able to, in their habitat, figure out where there is more food potentially. Are they putting their trunk down into an anthill? Like, what are they sniffing out, and why do they need to know whether or not there's more of it?
Yeah, so that's a really good question. I think one of the questions that has to come from this is how far this works, what the distance of their abilities would be, because elephants will walk hundreds of miles, thousands of miles across Africa following rainfall, following their food, because most of what they eat is vegetation. So if they can sniff out the rain, I mean, we all know that rain smells a particular way, but if they can smell the areas that have the heaviest growth, for example, then that might be really advantageous for them to know which direction to walk in all day to cover this vast kind of path to food, because otherwise they're just kind of going, okay, the rain's roughly over there, I'm going to walk over there. But if they know, oh, there's a giant patch of grass, if we walk northeast instead of just east, that could make a huge difference for the survival of their herd.
Yeah. So they're kind of like moths, except instead of pheromones that they're catching on their very sensitive antennae, they're sniffing out the scent of rain or other olfactory sensory stimuli that tell them which way to go in their environment.
There's a whole nother possibility, and that the use on food is completely kind of a byproduct. And actually what they need to be able to quantify via smell is the amount of a hormone. How kind of
mating hormones, aggression hormones. Yeah, maybe scent is a huge part of it.
Are there more females over there? I'd have a better chance of sneaking in and being a sneaker male over there. Like, there's
now I'm imagining elephants sneaking in on tiptoes.
Yes.
Me too. Imagine a ninja elephant. A ninja wearing an elephant suit.
They'd get up real close and then they'd go, psst, psst, come over here.
Elephant, you scared me. You snuck up on me so stealthily.
Oh my goodness.
Stealthy elephants.
Yes.
There we go. Thank you, Blair. Everyone, it is time for us to take a break. We have come to the end of the first half of our show, and we'll return in just a few moments with more stories. I got blood in the second half of the show. We'll be back in just a few more moments for more This Week in Science.
Can't explain things you've heard more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need.
Thank you for watching or listening to This Week in Science. We appreciate that you have spent part of your week with us. To keep this show going, we do rely on support from audience members like you. So I ask that you head over to our website, TWIS.org, where you can find information about how you can help our show out. Number one is just clicking on that big orange subscribe button. Click subscribe. It'll help you. You can choose whether to subscribe YouTube, iTunes, or Google, and then you'll be all subscribed. You can tell people to go to TWIS.org and to subscribe if you think this is a wonderful podcast. Additionally, we have a Zazzle store. Our Zazzle store is where you can find all the TWIS goodies, t-shirts, polo shirts, tote bags, pillows, wonderful things, TWIS logos all over them.
Phone
Art
cases.
Right. Art from Blair's Animal Corner calendars of years past. And a portion of the proceeds supports This Week in Science. Back at TWIS.org, you can also click on the yellow donate button on the side that allows you to choose one-time or recurring donations to This Week in Science through PayPal. We also have a link to our Patreon community, and Patreon is where crowdfunding takes place. That's right. You can click the Become a Patron button and then choose the level of your support. You will be charged this once per month, $10 a month and more, and you will be thanked by name on the show. So get in there. Patreon, PayPal, support this podcast. Independent science media coming to you every single week. And if you want more This Week in Science, this weekend, we're going to be in Santa Fe, New Mexico at the Interplanetary Festival. At the Interplanetary Festival, you will be able to find other wonderful podcasts on the podcast stage, but we will be podcasting June 15th, this Saturday, 4:15 p.m. Mountain Time. We will do our best to live stream to you through our YouTube channel and twis.org/live. But if we're not able to do that, we'll be recording the podcast and sending it to you through our podcast channel. Oh, we couldn't do any of this without you. We thank you so much for your support.
Can't explain the things you've heard more than intuition. A lot of reason shows the way to go. New conclusion. The methods are hot.
And we're back. You're listening to This Week in Science. And what time is it, Kiki?
Oh my goodness, it is time for This Weekend. What has science done for me? Okay, and since Kai is our guest this week, I'm going to throw this out to Mr. Kai. What has science done for you lately?
Well, science lately has... I'll go last.
No,
must.
Tell us what does it let you do in your life?
Well, it lets me I did my animal report.
Oh.
Thank you, Blair, for giving me the idea of doing snow leopards. I did it, and I did as good as I could, and it was actually pretty good.
That's awesome. What did you learn about snow leopards, or how did you learn about snow leopards, since we're talking about what science
One
did
thing
for us
that
lately?
I searched up to learn it was this one place called... Well, I just searched this up on Google. Well, yeah, I searched up 15 amazing facts about snow leopards. I copied out the facts and I changed them up, and I changed up the facts about snow leopards a bit, and then I changed with them and then used them inside my book.
In your report?
Yeah.
Right. You wrote them yourself, right?
So Kai, when I had to do animal reports when I was in school, it was a little bit different. So I feel like science has helped you a lot, because I would have to go, my mom or dad would have to take me to the library. I would have to go find a little card file, find a little card with a number on it. Then I would have to go find the book. And then I would have to check out a bunch of books, take them home, read through them, copy things out. I got very excited when I was in high school and I got some CD-ROMs that had an encyclopedia on them. That was like a very special day because I didn't have to go to the library anymore, which the library is still very fun, but it's pretty cool that you can learn 15 snow leopard facts all in one place just like that. That's awesome.
Yeah. Did you go to the library?
No, I didn't have to.
Oh my gosh. Do you remember any of the snow leopard facts that you really liked?
Well, my favorite one was probably if they walk in really deep snow, and they kind of don't like sink into the snow. They kind of just float on top of it in a way.
Yeah. Why is that? Do you remember why?
They have really furry paws that spread their weight more evenly across the snow so that they don't just sink down, and they help isolate the sound of their movement.
Yeah, they have these huge feet that are like snowshoes, and so they can spread out their weight. So awesome. Nice.
That's why we have snowshoes. Yeah. To be able to spread that out. We have to strap things onto our feet to keep us from...
Well, snow leopards can just walk outside.
Snow leopards just get to do that. Yeah.
They're not as graceful as the snow leopard.
Yeah, and Kai watches all sorts of YouTube videos about science, and so I would say that technology and science come to him all wrapped up in a nice little package.
Yep.
All the time.
Probably my favorite videos to watch. shout out if you're watching this. Well, probably shout out to this YouTuber who he's really good at animating. I'm surprised they actually animated so well. Cruzgat.
Oh,
well, I don't
Kurzagat.
know how to say it. I don't know how to
Yes.
pronounce it.
Yes, it's a good account.
It's in a nutshell.
It's
in a
German.
nutshell.
It's German for in a nutshell.
It's really, really good.
That's
They talk about like
fun.
everything. They talk about lots of stuff like TWIS. Basically
awesome.
an animated version of TWIS.
Nice.
Very cool. Thank you, Kai,
You're welcome.
for sharing what science has done for you lately. It helped him with his report. If any of you are working on reports or whatever, you have little notes for us about what science has done for you lately, send us a note. It doesn't have to be a crazy thing. It doesn't have to be long and involved. Just let us know what has science done for you lately. Send me an email, kirsten@thisweekinscience.com, or leave us a message on our Facebook page. That's facebook.com/thisweekinscience. All right, Blair, you got a story for me?
Yes, I'm channeling Justin tonight by bringing a nice microbiome story all about the gut. This is all about specifically the guts of twins. This is the largest ongoing scientific nutrition study of its kind. It's an international team of leading scientists from King's College London, Massachusetts General Hospital, the nutritional science company Zoe, Z-O-E, and more. And they showed individual responses to foods that are unique to every individual, even between identical twins. So this long-term study was specifically trying to figure out dietary guidelines and if there is any potential to a one-size-fits-all guideline for diet. And I think this could be extrapolated. I don't know if this is exactly what this team of research were thinking about, but I'm thinking about things as simple as the 2,000 calorie a day guideline. So there's all sorts of guidelines that we have that we use as kind of the baseline for what a healthy diet is for a human being. And the more we're learning, the more specific it is to each individual human. So these one-size-fits-all dietary guidelines have now proven to be too simplistic. There is a personalized approach that is necessary to nutrition in order to provide long-term health benefits. So the researchers measured blood levels of markers like sugar, insulin, and fat change in response to specific meals. And they looked at that along with data on activity, sleep, hunger, and their gut microbiome in thousands of participants in the US and the UK. Most of their study were twins, which is an amazing opportunity to be able to rule out genetic reasons for dissimilarities between these individuals. So the kind of key to this huge study was that they were able to use a machine learning technique from this company, Zoe, to look at all of this nutritional data. And they're actually taking this to try to make a test and an app so that people could then do a lot of their own testing on their own in order to maintain health and diet and stuff like that. This is a 25-year investigation in 14,000 twins. This is a big data set. They found that genetically identical twins had very different food preferences and responses. So some highlights of their findings. In 1,100 UK and US adults, 60% of these were twins. They looked at 2 weeks of regular blood sugar monitoring of glucose, insulin, fat levels, triglycerides, fat levels, and other blood markers to look at standardized and freely chosen meals. They had a variation in responses to the same meals, whether there were carbohydrates or fat in them. Some had rapid or prolonged increases in blood sugar and insulin, which experimentally are also related to weight gain and diabetes. Other had fat levels peaking and that lingered in the bloodstream hours after the meal, which is a pretty big difference from one person to another. This large variation only partly was explained by genetic factors. Less than 50% for glucose, less than 30% for insulin, and less than 20% for the triglyceride fluctuation. So there was only a weak correlation between an individual's response to fat and carbohydrates and their genetic makeup. This is the one that really blew me away. Identical twins who share all their genes, of course, and most of their environment, who are raised together, had different responses to identical foods. They found that identical twins shared just 37% of their gut microbes. Two unrelated individuals share about 35%. So in this specific study, the genetic makeup of these people had very little to do with their microbiome.
So this is something that we've talked about for like a lot of times. This is the microbiome is something that gets passed on to you when you're born. It potentially begins when you're in utero, and that it gets passed on to you while you're born and through breastfeeding or through life experience, and antibiotics can have an effect on it. And so there's all these environmental impacts to what your microbiome actually is. And
yeah, so I feel like we keep going back and forth a lot. We'll have a study that is exploring genetic links to microbiome. We'll have I feel like mostly it's been something else. It's been environmental stuff. But what one of the researchers from Harvard Medical School, a gastroenterologist there, kind of chimed in on this, is that it's actually super reassuring because it shows that the genetic makeup only partially explains how our body responds to food, which means our metabolism is not fixed and we have the power to change it, which also means we have an avenue to tailor our diets to the bacteria in our gut, which helps us metabolize nutrients. So I think before, we've talked a lot on the show about changing up your microbiome. This is kind of a completely different, this is turning that on its head and saying, identify your microbiome and adjust your diet to that. So I think neither way is perfect, but both are probably part of a perfect symphony of taking care of your body. If you can recognize, I mean, this is something that there are people that are gluten intolerant. There are people that are allergic to gluten. There are all these different kind of levels of gluten sensitivity that are, often the butts of jokes these days. But what some of this might be is that you have a particular microbiome that doesn't process gluten well. That doesn't mean you can never eat gluten again, that you have celiac disease or anything like this, but it might mean that it's a food that you might want to avoid, just like there are people who are lactose intolerant, right? So like, if you can identify that you have foods that don't make you feel good or that spike your insulin levels, that would be a good thing to know. And if we can figure that out from a nice microbiome swab, that would be great.
Yeah, I mean, the one thing this is research that's done by scientists. These are, these scientists have great credentials, but they're also, this is a company. Zoe is a company that wants to sell a product, right? And so there is this small bit of my mind that goes, well, this is a company that wants to, I mean, there are many companies nowadays that are using science-based reasoning or design behind their products. And so it's not necessarily that there's anything wrong with what they're presenting, but I do think that we need to also look at this with a grain, a little grain of salt. Well,
yeah. But, I think that's a very good point. I think that what something you could take away from this, though, is that in the future, part of holistic health care from your general practitioner will be looking at your microbiome.
Yeah.
And I think that's pretty clear.
Yeah. And I think your point also is, is your allergy or your intolerance, is it genetic in nature or is it because of your microbiome? And if it's because of your microbiome, is that then something that you can change? Can you take probiotics or prebiotics that might help influence your microbiome? Can you do fecal transplants, whatever they happen to be, to influence your microbiome to be a different state, to change that?
Yeah. Like some
Intolerance,
people can take their little lactose eating enzyme booster before they have their ice cream, and then they don't feel so crummy afterwards. If this is something where I can know, hey, bread makes me bloated, it makes my insulin spike, it makes me feel terrible, I'm going to pop a little booster, a probiotic pill that will help with the process of this bread that I'm about to eat. That would be great. I'd be so into it.
Yes. Yes, let us eat all the foods that we want to eat without the gaseous byproducts.
Yeah.
No.
Or we could just skip straight to the poop pills. Honestly, sign me up.
Or just Beano, whatever. Gas X, Beano. Yeah, let's talk about blood. Let's move forward from the gut microbiome to blood. So we have talked on the show many times about the shortage of blood for transfusions. Anyone who has given blood knows that they are giving to a very good cause, helping people to survive in all sorts of situations where if they did not have that blood, they would not. Now, Blair, you are a universal donor. Is that what I remember? No.
I am the universal recipient. So my blood is almost useless.
You can take them all.
Yeah. I can only give my blood to other universal recipients.
Right. Well, what if something could change that?
Yes.
Our blood molecules have these sugar molecules on the outside of them that are very similar to molecules called mucins that line the inside of our intestines. And so researchers from British Columbia trying to figure out, could we develop a system to take away those sugars that mark them as particular blood types and also basically influence the way that they can interact with each other and with immune cells and with the body? If we could change them or eat them away, could we, say, take type A blood and turn it into universal donor type? Could we turn it into type O blood?
Well, because type O blood is the one that's basically naked, right?
It's naked.
It has nothing on the outside. Yeah.
No antigens. It's got nothing.
Is that kind of bad though?
Not necessarily. It's good for being able to give blood, but in terms of how it interacts with the immune system, it's potentially not as good. The antigens are a protective factor. So there are pros and cons. But when people are dying because they don't have enough blood, if you can make more blood that everybody can have, then it's great. Like I'm type B. And so I can take blood from
What's my type?
Universal donor. I don't know. It's a great question.
Universal
donor.
You and my dad.
Or I can take from other type Bs. But I can't take from a type A person. I can't take blood from Blair. But what if I could? And so these researchers were looking at these mucins in the gut, and they found bacteria that eat these mucins, that have enzymes that break them down and allow them to do things in the gut. And so they started looking at these E. coli and started looking for enzymes that could do to blood what they do to cells in the gut. So the researchers found a bunch of enzymes. They found two that when they tested the one of them by themselves, it didn't really do anything. But when they put two of them together, cut off the sugars, there were no sugars that they could detect in their tests on type A blood, potentially indicating that they had turned type A blood into universal donor type O. Now, there are more tests that need to be done, but there are other methods of creating type O blood from other blood types. However, they're really expensive and not very efficient. This could potentially be our inexpensive, efficient answer. Bacterial gut enzymes for changing blood types.
So this is, you'd pull my AB positive blood and you'd put this stuff.
You'd basically have your bags of blood that have come from the transfusions, and then you would
put the gut bacteria in there.
Yeah,
shake it up real quick.
Right, shake it, a little blood cocktail. No, I'm imagining that it wouldn't be the bacteria because that's less efficient, but actually the enzymes. And so isolate these enzymes, manufacture these enzymes at scale, and then
I feel like you would probably run them through some sort of membrane, right? So you could you could select for only the blood molecules the blood cells that have been altered.
Right.
So you could potentially select for the type O blood that has been transformed, and then you would end up with this clean system where you wouldn't end up with any enzymes on the other side of this membrane. You'd only get the changed blood. I feel like that seems doable.
It seems doable, and it's very exciting. And then another exciting medical discovery this week that I saw this story, and it kind of blew my mind. One of the big issues with people aging is hardening of arteries, cardiovascular disease that very often leads to heart problems, heart attacks that lead to death. Researchers looking for over the last 10 years, basically doing just basic research into proteins and DNA and cells, were able to determine that there is a molecule called poly-ADP ribose, or PAR, P-A-R for short, and it's normally associated with DNA repair. And so when damage occurs inside the cells to DNA, PAR comes in and is part of the system that fixes it. However, they used to think that PAR only existed inside cells, but it turns out it also exists outside the cells too. And they've found that hardening of the arteries is the interaction of PAR reacting to damage, stress damage. So say you're smoking, or there's stress of some kind like dialysis, or something that has affected the way blood is flowing through your arteries, leading to cellular damage. PAR interacts with calcium and crystallizes, hardening the arteries. So now they have a mechanism for how artery hardening is occurring. But beyond that, they also found that a very commonly, widely prescribed antibiotic called minocycline, it's used to treat acne very often, can prevent the buildup of calcium in the circulatory system and remove and repair hardening of arteries. So this is a treatment that's already out there, approved by the FDA, very widely circulated, and people are able to they don't have bad reactions to it. And then secondly, they've got a mechanism. And so they're really going to start pushing forward to see if maybe this antibiotic can treat this cardiovascular disease.
So this is great. I think it's awesome. But I feel like this is like the third or fourth story I've heard in the past couple years about an antibiotic used for one specific thing turning out to do something completely different. It's always beneficial, which is great.
It's great.
But it also just sounds to me like we have no idea how our body works.
But this is also the interesting thing. It's like the way that antibiotics work is fascinating as well. They have these interactions with bacteria that prevent the bacteria from reproducing. But at the same time, these antibiotics interact with our bodies and our cells and the chemical processes that are taking place inside of us. These antibiotics, there's all sorts of stuff that they do. That's where side effects come from. That's where they're not just affecting the bacteria, the microbiome. Yeah.
Which is another reason to not overprescribe them. I feel like we're very good about recognizing the dangers about superbacteria and all these sorts of things. But this is just another reminder that any medicine or drug could have all sorts of, we're a complicated system.
We are a
There's
complicated
lots going
system
on.
for sure. I mean, the thing that I'm also interested in is, I mean, so many antibiotics are becoming, there's resistance in bacterial species. And so, all right, the antibiotics don't work to actually get rid of bacteria anymore, but hey, we can use it to stop your arteries hardening.
Yeah,
so at
that is
least
good
we
news.
have these potential tools that we can continue to use, just not for bacteria.
Yeah, sure. Good. Yep. That's great.
Yeah, we'll just put it in our little basket. That's right. Do you have another story?
Yes, I have one more story. It is a thrilling tale of feather lice and the witnessing of evolution in a very short amount of time. Parasitic feather lice, they are a very particular type of lice. They live on pigeon feathers, and they have some very interesting traits about them. One of them being that female lice are about 13% bigger than males. That's a sizable difference. And they, in particular, it is a size difference that can cause problems when it comes to mating. So basically, there's a size at which the male can still mate with the female. But at a certain point, if the female is too large, it's physically not possible. And so this kind of perfect storm of specialization was the perfect backdrop for researchers from Emory University to look at reproductive isolation. They started with a single population of parasitic feather lice. They split that population into two, transferred them onto different sized hosts, pigeons with small feathers and pigeons with large feathers. The pigeons preened the lice naturally, and populations adapted to evolve differences in body size to what was best on the different sized feathers. After 60 generations, which is just 4 years, the biologists saw larger lice on larger pigeons and smaller lice on smaller pigeons. They paired them to different sized male and females from either population, and when they did that, the females laid 0 eggs. This is reproductive isolation. Yeah, those are such weird looking animals. Hey, those of you listening, please go to TWIS.org and click on the show notes from tonight and check out the video of feather lice copulating on videotape. It is hands down probably the weirdest thing you'll see today. But it's also fascinating. So the female is much larger than the male, but the body size differences as a result of separating them onto different sized pigeons appear to be preventing the lice from physically mating with each other. This is speciation. This is the beginning of a new species. Reproductive isolation is usually the first step to what becomes a functionally distinct species. So, in order to find this out, they wanted to see why they weren't getting any eggs. And in order to figure that out, they wanted to film the lice attempting to mate. But this is where I just gotta love the narrative style of biological research, because you get to hear these scientists and the troubles they had trying to figure out how to videotape lice mating. They would not do it on videotape. Then just one day, an undergraduate researcher brought a heating pad into the lab for her sore back. And Eureka, the head researcher, had an idea. He used a hot pad, which was tuned to the bird's core temperature. So they tried to replicate how warm it is on a pigeon. And all of a sudden, bam, the lice started attempting to mate. But what they saw as a result was that male lice physically could not mate with females. So they figured that was what was happening. Physically, they could not reach over from the larger to the smaller or the smaller to the larger. So there is this kind of very specific size at which the female is about 13% larger than the male, where all of the puzzle pieces fit just right. But if the male is closer in size or farther in size than that very small margin, it's not possible. And so this is considered ecological speciation. That's when this was first championed by Charles Darwin, of course. This is when a ecological change causes a shift in adaptation that makes reproductive barriers. And so in this case, the different sized pigeons were enough of a barrier so that the lice didn't lay eggs.
This is all so fascinating. Just the way all of this is working together. So you have to fit in the space between the feathers to be able to not get picked off.
Mm-hmm.
And then you got to be big to have lots of eggs, but you can't be too big because then the males don't work with you. So you have to fit within this very specific set of parameters for size.
Yes. It's a reminder that life persists because of so many random changes that eventually one works. So everything has to be exactly right, and it only makes sense if you have a million iterations of a thing and eventually one thing fits. And then it has to fit over and over again so that you have enough genetic diversity to actually make a species, which is
mind-blowing.
There it is. Yeah. Speciation in 4 years.
Genetic diversity. Mind-blowing. We'll see. So will they be calling it a new species? We will find out.
Yeah, that is a great question. Honestly, based on my previous conversations about species, I don't really care. But I feel like identifying species is important in one case, and it is protection of a species. Other than that, It's just nature doing all this crazy stuff. Sometimes it works, sometimes it doesn't. Sometimes they curl back in on each other, like in hominids. Like, it's just whatever, man. It's just us trying to define things. But really, what's interesting
because we have to put everything in our boxes.
Yeah.
You must fit in this little box. Never mind what you're actually doing. You have to fit our characterization.
Yeah. I think if I were to kind of rebrand the study, I would say ecological reproductive barriers in 4 years.
Nice. I like that.
Yeah.
There we go. There we go. Not necessarily a species, just barriers.
Yeah.
Like building a wall. I didn't say that. But
I mean, if nature did it.
Right. But since I did say that, what other political decisions are affecting our science? Recently, the Trump administration has put a ban in place on fetal tissue research within the National Institutes of Health. June 5th, yeah, there was a 9-month review led by officials at the Department of Health and Human Services. And it issued a decree on this policy, and it has 3 components. One is that there is a contract between the NIH and UC San Francisco, and UC San Francisco was using fetal tissue to develop humanized mice for HIV drug testing to create a model with which we could actually start to create better drugs to stop the HIV epidemic. That partnership is done. The NIH, there is no scientist within it allowed to do any research using fetal tissue anymore. none, nada, zip, zilch. And the third aspect of this is that there is a longer review process in place for anyone applying for funding from the NIH to do research that involves fetal tissue. Now, you might wonder why this is such a big deal. Well, humanizing mice and using fetal tissue for research does allow certain studies to take place that cannot be done in any other tissues. For example, study into human development. What happens during the time of fetal differentiation? You can't study that without fetal tissue. And if you want to know about humans specifically, you need human fetal tissue. So historically, going back a little ways, the NIH director, Francis Collins, in 2018 had noted that the agency was putting up $20 million over 2 years into research into alternatives to fetal tissue, but you have to have fetal test tissue to test the alternatives against. Otherwise, you have no way to determine the validity of those alternatives. And so at this point, with no fetal testing being allowed and the longer review time, what we're going to see are researchers just abandoning their research, which is what some people wanted to have happen. But it will stall a lot of research taking place into human health and treatments for disease.
What kind of policy is this? Is it something that can be lifted once
Great question.
things change?
Yep. Yes, this is a policy. This is not a law. So it is something that if a different administration were to come in, or even the same administration were to review these considerations and come to a different decision, the policy could change.
Well, I guess call your congressperson.
Right.
Ugh,
Yeah.
this is frustrating.
Yeah, but it will hold back a certain amount of important research going on.
Just think about all of the what has science done for me lately. Submissions that we have had that involved somebody's life being saved, that in some part of the testing probably involved some of these labs.
Yeah, and some labs, there are researchers whose entire grants actually cover much wider scopes of research, but a part of the research relies on fetal tissue. And so they will be abandoning the fetal tissue aspect so that they don't compromise their entire grant. And so there's compromise that is happening. There's compensation that's happening. This is going to affect research moving forward in the United States. So for the time being.
There's a really good question in the chat room as well. Does this affect non-federally funded fetal tissue research?
It does not.
Okay,
this
so
is
that's
only
good.
This is only related to the National Institutes of Health and any research being done at the NIH or research that could be funded through the NIH or that very specific partnership between the NIH and UC San Francisco.
Okay.
So it's not,
I guess.
It's not affecting everything. Private research or different funding sources will absolutely continue, but this does start to limit the influence of governmental funding into this area of science. It's getting toward the end of the show. My son has gotten tired. He's moved over to the corner and put his head down for a moment. You're yawning there.
No, I'm so sorry. I'm so sorry.
You're yawning. Well, so a new study suggests that it may not be yawning just because you're tired. It may be that you yawn because your brain's getting overheated.
Oh,
what?
Yeah. So researchers.
That's kind of tired though.
Maybe your brain's tired and it's not thermoregulating appropriately. But researchers used a warm or a cold or a room temperature pack against their carotid artery to change the temperature of the blood heading to the brain. And then the researchers looked at how many times people yawned or didn't yawn, and then whether or not those yawns were contagious and who found the most contagious. What this study determined is that cooler heads were less likely to yawn and also less likely to pick up on somebody else's yawn. So it adds further support to the idea of yawning as a thermoregulatory mechanism.
Interesting. So we're pulling in cool air.
We're pulling in
straight to the brain.
Air and oxygen, and it's improving circulation, blood pumping. Yes. The yawning. That's the idea, that maybe yawning has to do more with temperature.
Is it Stella?
Yes.
So now that we're yawning, this last story comes about what's after yawning, right?
Yes, and after the yawning, we go to sleep. And that's what we're going to be doing here in just a moment. But another study, this one out of the NIH, if you like these, National Institutes of Health, very important research and organization for our government. Yes. Stop. It's too loud. You cannot make any noise. The researchers at NIH looked at, they have just published a study in the journal Diabetes Care and have been looking at how problems with sleep, irregular sleep, your going to bedtime and the number of hours that you sleep affect your health. They followed 2,003 men and women ages 45 to 84. These people were involved in the multi-ethnic study of atherosclerosis, MESA. They were studied for about 6 years to find out whether there were any associations between sleep and metabolic abnormality. People also kept sleep diaries, and they responded to questionnaires about their habits and their lifestyle and other factors. And the researchers looking at everything determined that, yes, indeedy, if you have messed up sleep schedules where you go to sleep late one night, early another, you sleep a lot of hours one night, not so many the other, you're more likely to have metabolic problems like lower HDL cholesterol, higher waist circumference, higher blood pressure, total triglycerides, and also fasting glucose, which these are all indicators of metabolic disease that can lead to problems like diabetes. And so the researchers suggest that maintaining a regular sleep schedule can benefit your metabolic health and can benefit you overall. And we know for a fact that circadian rhythms are very important. And we've talked before about shift workers and why shift workers seem to have health issues. This may be a link between that, these irregular sleep times leading to that.
Yes, and for those individuals who have to do shift work because of their profession,
like
the more
podcasting,
or doctors, nurses, things like that.
Yes, exactly.
Anyway, security guards. There's lots of people that work overnight or have varying schedules. The more we can learn about that, the better we can try to prevent future medical issues related to that if we know that those people are in those lines of work.
Yep.
It would be great if everyone could sleep from 10 to 6 every night, but
it would be amazing, but that is not the way the world works. It's not the way our society works. And it's even not the way that people work. I mean, what this is suggesting, though, is it's the irregularity that is the thing that affects things the most. So it's not the 10 to 6 aspect, although there is other research that does lend itself to this idea that nighttime, daytime, there is a difference there because of circadian rhythms. But it seems to be that it's the lack of a schedule, the irregularity that is the issue.
Mm-hmm.
I like schedules. Schedules are great.
Mm-hmm.
They're good. Like, hey, we just came to the end of a show.
We did it.
Yeah.
Pretty much right on schedule.
We did. Pretty much. Just a few editing issues here or there, in there, but it's all all good. It's all good. You know, we have come to the end of another episode, and I just want to thank everyone for listening. Thank you for listening. Thank you for being a part of this program. Thank you for sharing your week with us, sharing this time with us. Thank you to Identity 4 for helping to record the show so that I have a podcast to put out. Thank you to Fada for helping with show notes, social media, and our chat rooms. Thanks to Gord McLeod for helping with the chat rooms. And I would like to thank our Patreon sponsors. Thank you to Paul Disney, Richard Onimus, Ed Dyer, Andy Gross, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard, Brian Condren, Dave Neighbor, David Williams, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stephen Albaran, John Ratnaswamy, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Sue Dolster, Dave Wilkinson, Ben Bignell, Richard Porter, Noodles, Kevin Reardon, Christoph Zook, Narek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Suzuki, Drim DePoe, Greg Riley, Sean Lamb, Ben Rothig, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Edna Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you for supporting us on Patreon. If you're interested in supporting us, you can find information at twis.org. Click on the Patreon link, or you can help us out simply by telling your friends about TWIS. On our next show, we'll be in Santa Fe at the Interplanetary Festival, June 15th. That's a Saturday, 4:15 p.m. Mountain Time. We will try and live stream, but we'll be releasing the show as a podcast. We'll also be back again next Wednesday, live, twis.org/live. At 8 p.m. Pacific time, our normal time. You can join our chat room. But if you don't make it, don't worry, because we do have past episodes at YouTube and at our website, twis.org.
Thanks, everyone, for listening to our show. We hope you enjoyed it. TWIS is also available as a podcast. Just search for This Week in Science in iTunes, or TWIS for Droid in the Android marketplace, or you just go to any Apple area and search This Week for Science. We'll be there.
And for more information on anything you've heard here today, show notes are going to be available on our website, twis.org. You can make comments and start conversations with the hosts and other listeners there. That's pretty.
Or you can contact us directly. You can email Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS, in the subject line, or you are likely to be spam filtered into oblivion. You can also ping us on Twitter, where we are at TWIS Science with one S, at Dr. Kiki, at Jacksonfly, or at Blair's Menagerie. You can also check out at Shouty Blair. I've been very shouty lately. We love your feedback. If there is a topic you would like us to cover or address, or a suggestion for an interview, please let us know.
And we will be back here next week. We hope you'll join us once again for more great science news.
And if you've learned anything at all from the show tonight, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. Science. I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. Science. I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. Science. This Week in 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 Science.
And that brings us to the end of another show with some different kind of audio today. It was good.
Like Twitch screaming.
But we're on the air still, even though we're done. You did a good job, Kai.
Thanks.
Yeah, that was good.
Yeah, that was fun.
Oh, here's a good thing we could do.
It was fun. I'm on the air right now. But how do you have pen on your face? How did you too?
Oh, look, here's a good one we could do. Llamas could help beat the flu.
Right? Llamas could help us beat the flu. I believe we talked about that story on TWIS a while ago. I think Justin brought it. There's lots of good science news out there. So much.
Ooh, somebody forgot how to spell Blair's Animal Corner.
Blair's
Bla-blurbity-blurbs
Animal Corner. Bla-blurbity-blurbs Animal Corner. Do you have any questions for our trip this weekend?
Oh, I have so many and I can't remember any of them right now. Are we doing anything that requires me to have something besides flats and jeans?
No.
Okay.
It's going to be in the low 80s, apparently. So just make sure you're comfortable.
Yeah, I think I'm going to be wearing jeans with a little bit of ankle showing. And I'll bring a bunch of sleeveless shirts.
What is this, like Victorian England or something?
Yeah. Here's the problem. I don't really own any shorts anymore.
It's sticky. What did you put on? What did you find to eat?
It's pickle chips.
What did you eat? It's sticky and sweet. Is there something sticky and sweet over there?
No.
What did you eat?
Pickle chips.
Uh-huh.
It was just pickle chips.
I don't believe you.
Hey.
You have sticky stuff all over your face. The evidence says otherwise.
Oh my. I just tweeted out from Shouty Blair my latest piece of art.
Yeah. Oh, look at that beetle. Look at that. That's a cool beetle. What kind of beetle is it?
That's a Hercules beetle, I think. Yeah.
The Hercules beetle.
Very
cool. It is tweeted out there. So if there's anyone watching, you got to follow Blair on Twitter.
At Shouty Blair.
Yeah.
I also retweeted it on my real Twitter, Blair's Menagerie.
Mm-hmm.
Look, oh my gosh, darn it.
You know, Blair's Shouty Blair account, it shouts all the time.
Yes, she only shouts.
Shouty Blair only shouts, which is pretty awesome.
It's most of the time, I feel like that is unfiltered what's going on in my brain. Is Shouty Blair.
I don't know how how do you make a podcast happen while a child is on a jungle gym?
No idea.
This is something that probably needs to happen because sitting still is an issue.
Yeah. You could get one of those chairs that like does this.
Oh, I love those. I love those. Yes.
As if he
Flexible
wasn't doing that
seating,
already.
right?
It was already making so much noise.
No, actually, they're really quiet, but they're really good. They have them at school. They make no noise, but they go around in circles, and they're really nice.
Do you have one in your classroom?
We have like 5.
Is that for the people who need movement?
No.
For anyone?
Yes.
Nice.
What is the sticky stuff? What did you get into? What did you? That's not pickle chips.
It is.
Pickle chips aren't sticky and blue.
Oh.
Seriously, in like 20
Am I getting
minutes.
frostbite?
No, in 20.
Does frostbite taste good? I didn't. Look, it's pickle chips. Let me show you.
No, I know you got the bag of pickle chips. There was something else in there. What did you
Maybe
find?
I was biting this.
Nope,
I
you
was
found
biting
something somewhere, and you're keeping it from me.
I did not take secrets. I did not hide anything.
Candy secrets. Do you want that back?
Yeah. I did not steal anything.
So I am going to ask you this question, Kiki. What's Meow Wolf?
I can answer that too.
Oh yeah, Kai, tell me what Meow Wolf is.
It's an interactive art museum, and I'm pretty sure every week it changes.
That's very exciting. I bought tickets because I was like, you know what? I'm sure it'll be fun, whatever it is.
It's going to be amazing. Yes,
That's
it's
great.
interactive art. It's going to be fun.
Oh my
you
God.
You can go
There's
through
a
it.
Meow Wolf in New York.
No, there's
Yes.
not.
There's something like it.
No. Well, maybe something like it, but there's no Meow Wolf in New York. They just opened in Las Vegas, and also they're opening in Denver. Stop picking your teeth on camera. Go off camera if you want to pick your teeth.
Santa Fe,
quietly.
Denver, Las Vegas.
But
There
it's
it
interactive art. The thing that I think is so cool about the Santa Fe Meow Wolf is that it is backed by George R.R. Martin.
No.
Yeah.
Is he from Santa Fe?
I don't know. Yeah, he must be somewhere around there.
Quick
Yeah.
Goog on that. God, Google knows me too well.
You smell like pickle chips.
Because I was eating them. I had handfuls.
I heard you eating them.
Yeah, he lives in Santa Fe.
Maybe we'll run into him at dinner.
That would be amazing.
I wouldn't doubt on it. I wouldn't count on it.
I wouldn't
You wouldn't
count
count on
it.
it. How many
No.
people are there in Santa Fe? I'm gonna look. Santa Fe.
It's small, isn't it? I mean, it's
What if there's
probably
only 2
sprawling
people there?
now.
What if there's only 2 people in Santa Fe?
A little more than 2.
More than 2.
83,000.
83,000. That's still very small.
Yeah. Yeah.
Yeah.
Very tiny.
Sounds great.
Less than a tenth of San Francisco.
Yeah.
And probably if we go to, we can also probably lines won't be that long, unless all 38,000 people went to one place.
Right. Unless they're all going there. But they're going to be at the Interplanetary Festival, so we'll be able to go to Meow Wolf with no problem on Sunday.
Yes,
Yay.
I'm looking forward to that. I'm so thrilled that you planned some things because I started looking at what to do in Santa Fe and I kept getting lost and just kind of distracted or whatever. And I was like, I don't know. I'll figure it out, I guess. So you were like, we're gonna do this stuff. Do you want to come? I was like, yes.
I don't have to make the plans. I will follow the people.
Yes. It'll be amazing.
Yeah, I mean, I just figured Saturday would be festival stuff all afternoon, you know, because it's like it starts at 1 or something on Saturday afternoon. So maybe we can go find a cool breakfast or something on Saturday. And
Perfect.
maybe hit a museum if we want to, but I'm thinking or we could just stroll around and look at.
We could go to an Evolutionary museum. That'd be fun. What?
I don't think they have. I mean, we'll find out. We'll see. I don't think they have science museums in Santa Fe.
No.
We have to go to Los Alamos for the science museums.
Yeah.
No.
Or Albuquerque.
Yay.
But there's
Let's
lots
go
of
to
art
Albuquerque.
in Santa Fe. Art and
Yay.
food. Yes.
Oh yes, the Bradbury Science Museum is far away. It's in White Rock. It's the
There we go.
closest thing. Well, that's all right. There's plenty to do. We're gonna see some turquoise.
We have lots of turquoise.
I've heard there are hot air balloons, according to
You need to stop.
according to Grace and Frankie, one of my favorite TV
No, it
shows.
landed on the mixer. What?
According to Grace and Frankie, one of my favorite TV
Oh,
shows.
yeah.
There are hot air balloons in Santa Fe.
I imagine there are many. Yes, high altitude. We've got high altitude, very kind of flat plain desert, but there are hills nearby. Yeah, I'm sure it's gorgeous. Can't wait.
Yeah. Okay, so I'm
I do want to go to Los Alamos. I wish we had time to do that.
what is
What? Los Alamos National Labs?
Oh yeah. Oh man.
I wish I could make that happen. I wish. Next time we're in Santa Fe.
Yeah, I guess. I was describing to someone the other day, no shade thrown at Santa Fe at all. But one of my favorite things about traveling for TWIS is that I end up going places that I probably wouldn't have picked on my own.
Right.
And it is amazing. I love that.
Right. When are you going to pick Baltimore or Philadelphia or
Yeah.
Yeah, and it's
Even
been great.
I feel like I've always wanted to go to
And now
Denver, but
Santa
I wasn't
Fe.
like banging down the door of Denver to go. And then we went and it was lovely. And yeah, I mean, we were in Philadelphia. We were also in Phoenixville, which I never would have been in probably otherwise, which was so fun.
Right. That little tiny town.
It was beautiful with its drive-through liquor stores. So odd. Okay, so I'll bring my laptop. I'm going to bring my HDMI connector for my laptop. I will try to print out intro and outro scripts for us.
Oh, cool. Great.
I probably won't print the whole thing, but I will print, I'll print the intro and outro so we have those so we don't have to look too far for those. I'm going to set myself an alarm right now to do that. And
I'm going to try and get my stories printed out before Friday.
I'm going to try to do that too. So my stories are adjacent, but not directly relevant.
That's fine.
Okay.
Yeah. Yeah.
Like one of them is something fun that we could talk about the social implications at some of the festival mixers.
That's interesting. What?
Yeah, just about peer pressure, you know.
Ooh.
I have something about food preservation, which could be relevant to space travel, for instance.
Right.
I have a super armor.
That's good.
And I have, for the short stories, I have satellites that are like Legos. So that's fun. Did we need to do a sound sample for my fan?
Yeah, we should do maybe Identity 4. Identity may have already finished his recording. Probably got a quiet enough bit of time in there.
Mm-hmm.
At least a couple of seconds of quiet.
Yeah. It's just how my life works.
What?
It's just how I talk. It's just my nature. My nature as a kid to be loud.
Mm-hmm.
Yes.
But we weren't talking about you being loud. Her fan is loud.
Oh.
Yeah.
Oh yeah, I can hear that.
I know,
Mhm.
Kai, you're my
That.
biggest fan, but this is also a large fan.
Oh, that's really loud.
Yeah, recording is uploading. Awesome. That's cool. We're good. There's going to be, there will be points in the recording where I will be able to get a couple of seconds for noise. We're good.
Mm-hmm.
You know what? Just the way it is. It's just the podcast. That's what we did. Live recording. This is what we do.
It's 1,000,000 degrees.
So yeah, are you super hot? It's hot in my basement, which is rare.
Yeah, it's like a nice summer day.
How hot was it in San Francisco today?
Today, it got to 81, which isn't too bad.
That's not that bad. Yeah.
Yesterday,
San Francisco,
92. We got to 92.
Over.
I think.
No, it was like 95. Maybe my car yesterday said it was 102.
Yeah, yesterday it was 95 in San Francisco, and the day before that it got to 101.
Last night it didn't get below 80 degrees until after midnight.
Yeah, well, that last night, the past 2 nights, I haven't slept very well at all because it has been so hot.
Apparently, it is 81 degrees outside right now.
Oof.
It's after 10 o'clock at night.
Boiling hot in Seattle.
Boiling hot in Seattle. Yeah, we're having a serious heat wave right now, all down the western coast.
Mm-hmm.
Yeah, I can barely leave the house. When I left the house after Kung Fu, I could barely see it. Like, I was actually getting dazed and seeing weird things.
Yeah.
It was so hot.
I'm trying to teach him how to, like, just swim. I was telling him yesterday, I'm like, yeah, it's hot outside. You just slow down your walk and you just got to, like, swim through the air. You just kind of, you go through it. You just got to part the air in front of you and you just move through it and you're just hot and you just do it. You just
That's
go.
great. I love that. So of course my teen program started for the summer this past week. And so I have, on some days I have like 15 teens that are doing interpretation out in the zoo, and I'm checking in on them and all this kind of stuff. And you know how teenagers are. So of course, the past couple of days, they have just been so like irritable and grumpy. And like one kid yesterday, I was like, hey, have you had water lately? Do you need to refill your water bottle? I want to make sure you're staying hydrated. And he was like, I already did it. And he like turned around and stormed out. Somebody's grumpy because of the heat.
Oh my God, that's funny.
Yeah. I mean, teens are predictable. Oh,
what?
yeah.
I don't really drink that much water.
That's not good, Kai.
But
You
then
should probably
sometimes
drink
I
more
chug
water.
them.
Yeah.
On some days, I barely have water. On other days, I chug up all the water.
Gotta just drink your water every day. It's good for you. You've been drinking it tonight. That's good. Say no to yellow pea, orange pea, brown.
I've been having
No
that lately.
brown pea.
I've been having orange pea lately.
Uh-oh.
No,
That's not
gotta
good.
drink- That's terrible.
Orange-ish.
I need to take you to the doctor, or you need to drink more water.
Okay.
I found
Mhm.
Now.
I found a very funny, let's see, pea color meaning. I found a very funny one a couple weeks ago. Maybe this is it. It's from the Mayo Clinic. Is this the right one?
Oh, what different colors mean? Yeah.
Yeah, it was kind of snarky explanation.
There's a purple one. Is there a purple one?
Purple.
Yes, purple is one, but you have to go to the doctor.
Yeah, you should not have purple pea.
I won't.
Oh yeah, this is
That's
the
like
one.
absolutely no water.
It's like
And you're also like bleeding.
what is that?
You're
Sodium
bleeding.
sodium perchlorate pea? You should not be producing
Combined with blood and very low water.
Ew.
That's probably what purple pea is.
Let's see. What does
8
it
liters
say?
a day.
This says, clear. You've been drinking too much water. Cut back. Yellowish to amber. Typical healthy urine. Red or pink. You ate red dyed things. Or you have an enlarged prostate, kidney stones, tumors in bladder. Talk to your doctor. Orange. It could be just dehydration, but if combined with light colored stool, it could mean issues with their bile duct. Blue or green. Very rare. Most likely connected to something you ate. In rare cases, it could mean a bacterial infection. Dark brown, dehydration, diet related, or a side effect of certain medications, porphyria, or liver disease. Cloudy, dehydration, urinary tract infection, or a symptom of chronic diseases and kidney conditions. This is not the snarky one I was thinking of. But
that was real. That was not snarky.
That was not snarky at all.
That was dark.
That was accurate.
And also very dark. And gross. And very nasty.
Let's see.
You don't want to know?
I do.
I find it very interesting. Oh, this is the one. It says purple. There's no such thing as purple urine. Oh yeah, pale straw color. You're normal, healthy, and well hydrated. Transparent yellow.
No.
You're normal. Dark yellow. Normal, but drink some water soon. It's like amber or honey. Your body isn't getting enough water. Drink some now. Syrup or brown ale. You could have liver disease or severe dehydration. Drink water and see a doctor if it persists. Pink to reddish. Have you been eating beetroot, blueberries, or rhubarb? If not, you may have blood in your urine, which could be a sign of kidney disease, tumors, UTIs, or prostate problems. Orange. You may not be drinking enough water. Or you what?
Or you ate a knife.
Oh no!
No, that's awful.
Yikes.
No.
Or you ate something really sharp.
Let's see, blue or green. Oh, there's a genetic disease that gives your urine blue or green tinges. Foaming or fizzing. A harmless hydraulic effect if occasional, but it could show signs of excess protein in your diet or a kidney problem. See your doctor if it persists. If you have foamy pee, go see a doctor. There you go.
Good to know.
There's no such thing as purple urine. You heard it here.
Can I just tell you a weird syndrome that actually sometimes happens?
Yeah.
With people's brains? So sometimes when people are eating, they forget to stop eating and they eat the utensils with it.
Really?
And then like they have to get surgery. And once this someone found like forks and like spoons in someone's stomach.
I understand that with goats.
Yeah.
Not with people.
Goats need surgery. Goats need serious surgery.
Yeah.
How do they eat stuff like that?
So I think that's actually usually a myth. They don't actually usually eat tin cans or anything like that. But they can eat paper. They do eat
It's
paper.
just that they just seem to chew on everything.
They chew on everything. They'll chew on my jacket when I go over to the farm. Yeah. Absolutely. They'll bite zippers off of jackets. Yeah.
Wait, what?
Mhm.
What?
Yeah, you gotta watch them. I think
It's
everything.
really cute.
Goats
I
are
remember a long time ago. We had these we went to this like farm in
Mhm.
San Francisco, I'm pretty sure, that was really, really nice. And the goats there were really, really nice to us. Except one kid got rammed by a goat.
No.
Yeah, it was the most tutorial goat. But all the other goats were really nice and they didn't seem to chew on anything. Basically just fed them.
Yeah.
And one of them just stood still.
So goats are really, really, really smart. So I feel like sometimes they're trying to just see what they can get away with. Sometimes they're bored. Sometimes they're like, you have food. If I chew on your jacket long enough, maybe you'll give me some food.
Or sometimes they're just trying to be silly.
Yeah. Sometimes they might just be silly. Absolutely.
Do you think goats play?
Yes.
Yeah.
Like, are they playful? That would.
Oh, yeah. They're little troublemakers. There was a goat at the Jerusalem Zoo. I think his name was Jacob. He would jump out of his pen just so you would turn him and go, Jacob, get back in there. And then he would jump back into his pen. That's play for sure.
Mhm.
So, yeah, I think they do, especially since they will play kind of ram at each other when they're smaller, when they're learning how to defend their
Well, that's like
territory.
learning how to fight, right?
Yeah, but I feel like it's associated with play. I don't know. I think that's potentially an interesting question.
Right. I mean, play in all species is, for young, a method of learning adult behaviors.
Right.
Right.
Yeah.
Yeah.
Like you could say that things like tag and dodgeball are somehow related to learning primitive survival tactics for humans.
It just
Yeah, like aiming rocks and throwing them at your enemies.
yeah, it just those games just taught me I didn't like playing those games.
Yeah. Same.
One person in the over here says that goats climb on things.
They do.
They do.
King of the mountain.
Yeah. They love it.
Mhm.
Yeah.
I'm the king of the mountain.
At the zoo, when we put the little head harnesses that you see on dogs sometimes, we put those on the goats and walk them around, and they'll jump up onto benches and stumps and stuff. It's very cool. They love it.
Goats.
Yeah.
I can handle little baby goats. Adult goats are creepy.
Yeah.
I don't like them.
One thing that's
They're weird eyes.
The horizontal
Yeah.
pupils. Yeah.
Nah.
the horizontal pupils are weird. They look like they
Not a
have
fan.
band-aids.
I'm not a fan. I look in their eyes and I'm like, you're not like us. Goats.
Yeah. I guess that makes sense why they're a symbol of like the devil, right? Is because they have weird eyes.
And they have these crazy horns.
And the crazy horns and the cloven
horns,
feet. Oh yeah, goats.
Yeah.
Goats, the devil.
Yeah.
Devil envoys, that's right.
The devil of the earth.
What?
The devil of the earth.
Dave Shorty's bringing up fainting goats. Yeah, I was just
Oh,
talking
yeah.
about those
Oh,
with
yeah, that's
somebody
so
recently.
cute.
Yeah.
And like my dad had videos like they when they get scared their muscles kind of just freeze and then they're just solid and then they just fall to the side.
Do they
When they get
lose
scared.
consciousness? I don't know
I
a lot
think.
about them.
Yeah, I'm pretty sure they just
Yeah.
lose consciousness and they just
I don't
faint.
know. That's a good question. I
Yeah,
would
because-
call them fainting goats, but I don't know that
Right.
for
Do
sure.
they just fall over, or are they like opossums and they fully lose consciousness and play dead, or
Yeah.
are they just falling over?
I don't know. This is a very
Lying
interesting
crow.
question.
No, there you go. I'm happy to oblige.
We gotta find it out.
Yeah.
Do fainting goats really faint? This is the Google.
This is the thing this is why all of us need the internet.
Yeah. This is what science did for us today.
Look at that. Second result, Google search. Do fainting goats really faint? Technically, no. They do not lose consciousness, but they stiffen up and fall over.
Is it bad for goats to
Interesting.
faint? Can goats die from fainting?
Do you know, Kiki, since we were talking about blood types before, what Marshall's blood type is?
No.
Ah.
I said in the show that I'm type B, but I actually don't even really know if that's what my blood type is.
Yeah.
I don't know what my blood type is.
Interesting.
I'm pretty sure Daddy said my blood type is A.
I don't know. At some point, your blood, when you were born, was tested, and
Do they do that? Oh.
Yeah. They type you.
Oh.
And because I was pregnant at one point, blood test, and they're like, "You're type whatever," and I'm like, "Great." Yeah. And
I don't know what I'm gonna be.
yeah, but I have no recollection. I have a piece of paper somewhere that says what blood type I am.
What if my blood type changes over my years?
That doesn't.
What if blood actually changes though? What if that actually happens though?
As far as I'm aware, it doesn't.
It'd be cool if our blood types actually change.
Yeah.
But
I want to find out if they do change or not change.
Okay.
So
like aging, everything will start changing, getting weaker, changing. And like the sugars on it might start collapsing, and like a type A or a type H or a B or any of that would turn into a type O when they're older. When they're more older, they'll get closer and closer to a type O.
It would be like if that enzyme ran amok.
You're right.
It's interesting. It's like a Punnett square. I forgot. It's like something from high school bio that I totally forgot that I looked up while we were talking. That if you're type A, you either have 2 A's or you have an A and an O. And if
the first one that came up, do blood types change?
Yeah.
And then like the fourth one that came up is, do blood types change with age?
Very rarely an individual's blood type changes through addition or suppression of an antigen in infection, malignancy, or autoimmune disease.
Autoimmune disease.
Or with a bone marrow transplant. Now that makes sense because the bone marrow is involved in producing your blood. So if you got a transplant,
it would create a different type of blood.
Yeah.
Yeah, but don't they usually try to match you for blood? I guess for bone marrow, they're looking for a genetic match of a different type.
Yeah,
so
not just blood type.
Yeah, so worst case, they might go for a mismatched blood type, but the proper genetic match for the blood marrow.
Yeah.
Wait, can you close up on that? And I kind of want to see which one I'd probably most likely be like.
So it all depends on what Marshall is, because it all depends on what both parents are, really. So, so for example, I'm AB positive. So I can give either an A or a B to a child if I have a child. And so it all depends on what the father has. So I don't remember what Brian is. To ask him.
Right. And as Hot Rod says, yes, if you get bitten by a spider, you become Spider-Man.
Oh, yeah, there's that.
There is that.
Yeah.
Radioactive spider injects his own genes into your genes and turns you into a spider human. And then you end up turning, and then over the years, it'll actually start infecting your whole genome, and then you turn into an actual spider.
Oh. Uh-oh.
Spider Kai. Spider Kai.
Have either of you ever seen the comic strips where Spider-Man ends up talking to an actual spider?
No, that's funny.
They're really cute.
The
spider just keeps going like, and at what point do we wrap them up and suck out their insides? And he's like, no.
No, we don't do that. No, we stop the bad guys. We wrap them up. We stop the bad. No, but when do I get to eat them? We
don't. Great. So I'm going to meet you at the airport. Both of you.
Yeah, all of us.
Now, here's an interesting thing about Spider-Man, though.
Yeah?
It's time for bed.
He tried to remove his powers once, but then it failed, and then he ended up giving himself 4 arms and then increasing the speed of him turning into a spider. And then eventually he turned into a spider human, and then he was turning back and forth from human to spider.
What?
Whoa.
When did that happen?
In the TV shows.
In the TV shows. Interesting.
And the comic books.
And the comic books.
It was in 1997 Spider-Man.
Oh, so specific.
Oh, okay.
You know what? I'm gonna inquire further with the Spider-Man expert in my life, and I will Be very interested to hear about that.
Mostly into symbiotes.
The symbiotes.
They're really interesting.
Yeah.
All right, Kai, time to say goodnight. Say goodnight, Kai.
Okay, good night.
Good night.
Say goodnight, Blair.
Good night, Blair. Say good night, Kiki.
Good night. Good night, everyone. Thank you for watching. We hope that you get some regular sleep.
Yes.
And don't overheat your brains.
Mhm.
Have a wonderful, wonderful science week. We look forward to talking with you, hopefully streaming to you on Saturday. We'll be back here next Wednesday. I will be probably on an airplane when I'm supposed to do my Twitch stream this Friday, so probably no Twitch stream. And yeah, but I will do what I can. We'll see what we can do. So we will try to be there. Thanks, everyone. We will see you very soon. Bye.